This is a video to recap enzymes and metabolic pathways. We're going to talk about the structure of the cell membrane, different types of enzyme reactions, and the effect of inhibitors on enzymes. Okay, so we're going to start by looking at the structure of the cell membrane. We know from Nat 5 that the cell membrane allows entry and exit of molecules from the cell. We will probably recognise this diagram as well. We can still see that we've got phospholipids. And the other thing that the cell membrane is made up of are proteins. There are three different types of protein that we need to know about. So we're going to talk about pumps, sometimes called carrier proteins, and we're going to look at, again, enzymes in more detail in this topic, and then also know what pores do. Okay, so pores are there to allow specific molecules or ions to pass through the cell membrane. So you can see that here we have a high concentration of this specific molecule and it is moving to a lower concentration. Now, because we're moving from a high concentration to a low concentration, we don't need ATP for that process. We also need to know about pumps. So pumps, we say, are carrier proteins. And what they do is they bind to specific molecules or ions and they temporarily enable them to cross the cell membrane. So we can see here that we've got a low concentration of this molecule and we've got a high concentration here. So because of that, we need ATP in order to phosphorylate this reaction. So pumps, they're to bind to specific molecules or ions and they temporarily enable them to pass through the cell membrane. And you will build a little bit in that knowledge when you do advanced higher. You'll talk about sodium, potassium pumps, but for the time being, that is all we need to know. And then the last one that we need to look at are enzymes. We are going to talk about enzymes in loads of detail in just a second, but we need to know that enzymes are there to catalyse a specific reaction. We'll look at, for example, we'll look at ATP synthase when we start to look at the process of respiration, and that is there to catalyse the production of ATP. So from Nat 5 biology, we remember that enzymes are required to speed up the rate of a chemical reaction. And what we're trying to do in an enzyme reaction is we're trying to convert substrates into products. And we need the help of an enzyme. So we've got our enzyme here and it has an active site. So that is the part that the substrate binds to. It's got an active site that is specific to one type of substrate. So this substrate starch is going to bind to the active site of that enzyme and it is going to break it down into smaller products. Now we call this a catabolic reaction. We're breaking down a larger substrate into a smaller product. Now, when we're doing this, we are breaking the bonds in this molecule of starch. And I think of bonds like a big elastic band. When you snap it, it releases energy. So we need to know that catabolic reactions release energy. The second type of reaction that we're going to look at is an anabolic reaction. So opposite of the catabolic reaction, we're looking at this enzyme here. And this time we are taking smaller substrates. And again, they're going to bind to the active site of that enzyme. And we're going to build it up into something bigger. Now, because we are having to build up all those bonds this time, we say that anabolic reactions require energy. Okay, so catabolic reactions release energy. We're taking a large substrate, breaking it down into a small product. And anabolic reactions, we're taking small substrate, building it up into a big product that requires energy. So when we're thinking about metabolic pathways, we're thinking of several enzyme reactions happening one after the other. And we can see that here we've got 4 different enzymes and we've got various products being made along the way. we need to know that each step in this pathway is catalysed by a specific enzyme. And we've got reversible steps, we've got irreversible steps, and we've got alternative routes as well that we can take. Okay, so we can see for this first step, we've got a reversible step, and that means that once A has been converted into B, we're also able to convert B back into A, and that can be quite useful. So let's say if we had B increasing to a really high concentration, we could convert it back into A, and that would restore balance inside the cell. And that's really useful as it prevents products being made when they're just not needed. Now we can also see once B has been converted into C, it doesn't matter how high that concentration of C is going to get, we cannot convert it back into B. So that's like the point of no return in this metabolic pathway. And the other step that we've got here is we have an alternative route. Now that could be quite good if, for example, let's say we had a mutation that occurred in enzyme 2, so this enzyme was no longer working. We wouldn't be able to convert B into C, but because we have this alternative route, we're able to miss out this, this series of reactions involving enzyme 2 and enzyme 3, and we're able to go straight from B to E. So we've got an alternative pathway that can occur. So this diagram is showing a process called induced fit, and we can see different stages in an enzyme reaction here. We're going to start by looking at the substrates. So it says that the substrates have high affinity for the active site, and what that means is they have a chemical attraction to the active site and they are going to move towards it. Now if we compare this enzyme at the start of reaction compared to here when the substrate binds, we can see that the active site has changed shape slightly. It's kind of moulded around those substrates. That is induced fit. So when the substrate binds, the active site changes shape slightly and it brings those substrates into closer contact with the active site. Think of it like a rubber glove around a hand, it's giving it that really tight fit. We're increasing the chance of the reaction taking place, or we could also say that we are lowering the activation energy, and that is just the energy that is required to break the bonds and change those substrates into products. And that's really good for our cells as they can't tolerate really high temperatures, so that enables reactions to occur at lower temperatures in the body. So once that reaction has happened and we've converted the substrates into products, we then say that those products have low affinity for the active site and they move away. Okay, so just looking at the effect of increasing substrate concentration on the rate of reaction, we need to be able to describe why we get an increase in rate of reaction when we increase the number of substrates. So at the start of this graph, we have very low enzyme activity, and the reason for that is we only have a couple of substrates involved in this reaction. So they're only able to bind to a couple of active sites, and they're only able to make a little bit of product. We've still got all of these other active sites free. Now, as we add more substrate, again, more substrate are able to bind to those active sites. Again, adding more substrate, binding to more active sites, and we're going to be getting more product. Therefore, we can see that the rate of reaction is increasing. Now, at this point, it's starting to level off, even when we add more substrates. So we've added loads and loads of substrate here. We're still increasing the substrate concentration, but there are no more active sites for them to bind to. Therefore, we have reached the highest rate of enzyme activity here. All the active sites are involved in the reaction. It doesn't matter how much more substrate we add, we cannot increase the rate of reaction any further. So now we're going to look at enzyme inhibition. There's two ways that enzyme activity can be inhibited or rate of reaction can be decreased, and we can use competitive inhibitors and we can use non-competitive inhibitors. And both of these inhibitors decrease the rate of enzyme activity, by preventing the substrate from binding, but they do this in different ways. So the first type, the competitive inhibitor, if we look at the inhibitor here, you'll notice that it has this similar shape to the substrate. And because of this, what it is able to do is it is able to bind to the active site and prevent the substrate from binding. This is going to decrease the rate of enzyme activity because less substrate can be converted into product. The second type is a non-competitive, so you'll notice that if we look at this inhibitor here, it has a completely different shape to this substrate, and for this reason it can't bind to the active site. We say it binds away from the active site, or you can call this an allosteric site. Now when that inhibitor binds to that or binds away from that active site, you'll notice that this active site changes shape. So that substrate can no longer bind to that active site. Again, rate of reaction is going to decrease. Okay, so what we should be able to do for our exam is look at a graph like this, and we should be able to describe what is happening. We should be able to identify which line is showing which type of inhibitor just based on the results, based on the rate of reaction. So starting by looking at this line that is showing the enzyme only, or the control experiment, we don't have any inhibitor here. So we can see that as we add more substrate, the enzyme activity is increasing. So again, adding more substrate, it's binding to those active sites until it reaches a point where all of those active sites are occupied by substrates, and we start to level off. We cannot convert any more substrate into product. All of the active sites are occupied. Okay, so now we're going to look at the one that has the competitive inhibitor in it. And you will notice that as we increase the substrate concentration, the rate of reaction does increase. Now, just a reminder of what the competitive inhibitor is doing. So remember, your competitive inhibitor has a similar shape to the substrate, and therefore it is binding to that active site. Now, that means that at low substrate concentrations, we're only going to have a couple of substrates competing with those competitive inhibitors for the active site. But the more substrates we add, the more chance there is of those substrates binding to that active site. So what we see is this increase in enzyme activity, or we say we are reversing the effect of that competitive inhibitor, because the more substrate you add, the more chance it has of binding to those active sites. We can still see that the rate of reaction is a little bit lower than the enzyme-only experiment because there's still some inhibitors that are going to be binding to those active sites. Okay, so lastly, looking at the non-competitive inhibitor, and remember, non-competitive inhibitors are binding away from the active site and they are changing the shape of the active site. So that is key here. So starting off by looking at really low substrate concentration, there's my substrate and it's bound to this active site. We've got a little bit of enzyme activity, or we're seeing a slight increase in rate of reaction because we've converted some of that substrate into product. Now, the problem with the non-competitive inhibitor is, again, they have a different shape. They're binding away from the active site and changing the shape of the active site. Again, binding away, changing the shape. Now what that means is, it doesn't matter how much substrate I'm adding. So at this point here we have really high substrate concentration, but enzyme activity is still really low. There are no active sites available to bind to because all of them have been changed shape by that non-competitive inhibitor. So key things for your non-competitive inhibitor: enzyme activity is always going to be low, or it is not reversible. Whereas your competitive inhibitor, as we add more substrates, they're able to outcompete that competitive inhibitor. They're able to bind to more active sites, and therefore we say that we can reverse the effect of that competitive inhibitor. So the last thing that we're going to look at is feedback inhibition by an end product. Now what we can see here is a metabolic pathway that involves converting metabolite A into metabolite D using 3 enzymes. Now, metabolite D in this case is our end product. And what has happened in our cell is we say it has reached a critical concentration, or we've made too much of it. Now, when this happens, this end product is going to act as an inhibitor, and it is going to go back and it's going to inhibit an enzyme earlier in the pathway. In this case, it's inhibiting enzyme 1. So this conversion can no longer happen. We can't make A into B, and therefore this metabolic pathway can't continue. So what we say is we prevent further synthesis of that end product. Metabolite D is going to no longer be able to be made, and therefore we're going to decrease it. And that is really good for our cells because it prevents a product being made when it is not needed.